All right, so we are moving on. We're going to do some cellular respiration. Make sure that you watch this crash course that is embedded in here. Sometimes if I can't get the information out clearly, it's great to hear it from another voice. So we have crash course to begin this PowerPoint, and there is Bozeman to end it. This is a complicated subject, so the more voices that you can hear it from, the better. And then you guys need to add your own voices. So make sure in either a study group or with your parents that you vocalize this information, that you talk it over with someone. So cellular respiration is defined as the controlled release of energy from organic compounds in cells to form ATP. What does this mean? You're pumping in glucose, that's your organic compound, into the mitochondria, and you are getting ATP, which is our chemical energy from that mitochondria. Covalent bonds are slowly oxidized by enzymes, and this oxidation releases ATP molecules. Takes place in the presence or absence of oxygen, which means we can do this without oxygen anaerobically, but we're not going to get a lot of ATP. The best use of our glucose is to do it aerobically, and then we get a good amount of energy from it. You need to know these numbers over here on the side. So this line right here represents crossing over from anaerobic to aerobic. So glycolysis is an anaerobic process. It takes place in the cytoplasm. We don't need any oxygen for it, but we only get 2 ATP from the process. This Krebs cycle and electron transport system, all of this is in the mitochondria. It needs oxygen because oxygen is the end receptor for the process. And out of it, you're going to get 34, maybe 36 ATP. These numbers aren't exact, but if you're asked on a test, these are the numbers you need to give. So sometimes our bodies are more efficient than others. Some organisms are more efficient than others. Sometimes we get more ATP or less ATP than this. But in general, on average, these are our numbers. So glycolysis is the first step. We take this really big glucose molecule and we jumpstart it with 2 ATP. At the end, we end up with 2 pyruvate. So each of them are a half a glucose essentially, 4 ATP and 2 NADPH. Now, when we talk about glycolysis, remember in the last slide, it had 2 ATP as your net gain because, yeah, we get 4 at the end, but we had to add 2 to it. So our gross gain is 4, but our net, what we're actually getting out of this is 2 ATP that we can use. The breakdown of 1 molecule of glucose into 2 molecules of pyruvate with the release of some energy. This is taking place in the cytoplasm of the cell, and it is anaerobic. It doesn't require oxygen. Guys, anything in blue on this PowerPoint, you need to pause me, watch the video. Alcoholic fermentation is what you guys are going to do in your lab starting on Monday. Alcoholic fermentation done when no oxygen is present starts with glycolysis, and then we take that pyruvate and we're not doing anything with oxygen. We're not doing a full Krebs cycle. We're just going to push it through the system again and squeeze out a little bit more energy. So the yeast turn the pyruvate into ethanol and carbon dioxide. Ethanol is an alcohol. Carbon dioxide is the gas that's given off. Both of those are byproducts. And we're going to use that ethanol to make alcohol and the carbon dioxide to make your bread rise. Lactic acid fermentation is what's done in animals. So cells that normally do aerobic respiration can perform lactic acid fermentation as a last-ditch effort to keep you alive, to keep you running, to keep you from getting yelled at by your coach. Aerobic cellular respiration, on the other hand, is done in the presence of oxygen. We take the pyruvate and we put it into the mitochondria, and then we break down those bonds, and in doing so, we yield 28 to 34 more ATP. That's all the SL stuff. Now remember, if you are going to sit the SL exam, which the majority of you are, come May, that's all you need for the May exam. However, for all of my exams, I teach it at the HL level because you can never be too smart. And so here's the added HL cellular respiration. So we're going to go back and revisit some of that stuff that we just kind of skimmed over a second ago. First thing we need to do is remember some chemistry. Oxidation and reduction. This is a redox reaction. It's enzyme-controlled transfer of electrons. This is what's going on in photosynthesis. It's what's going on in cellular respiration. This is what's generating the majority of the energy that we use. So you may have heard of oil rig before in your chemistry days. Oxidation is the loss of electrons. It involves gaining an oxygen or losing a hydrogen, and energy is lost in an oxidative reaction. It's exergonic. It's giving off energy. In respiration, glucose is oxidized to carbon dioxide. So all the hydrogens are gradually removed from the glucose molecule. Reduction is gain of electrons. It involves losing oxygen or gaining hydrogen. It's endergonic. We're absorbing energy. We're making bonds. This substance now has the power to reduce other substances and become oxidized in the process. In respiration, oxygen is reduced to water. So oxidation is loss, reduction is gain. Here it is in table form. So comparing oxidation to reduction, you might be asked to do that on an exam. Just recreate this table. So glycolysis, the first step, we take our glucose and we do a linear series of reactions to break it down into 2-pyruvate. Again, this is taking place in the cytoplasm, and here's the chemical reaction. It's anaerobic, so it doesn't require oxygen. And glycolysis takes a 6-carbon hexose sugar and creates 2 3-carbon monosaccharides called pyruvate. The first step in this is phosphorylation. Now, phosphorylation is a big, fancy, scary scientific word. It essentially means adding a phosphate to a molecule. So we are going to phosphorylate ADP into ATP. Be aware this term phosphorylation, photophosphorylation, chemophosphorylation, you see it a lot. Take a breath, ignore the scientific jargon, and just realize anytime we phosphorylate something, we're adding a phosphate. 2 molecules of ATP are consumed for every molecule of glucose at this point, but you are generating 4 molecules of ATP, so you actually get to take home or use 2. The first step in glycolysis is lysis. Lysis means to cut. So fructose biphosphate is split, producing 2 triose phosphate molecules, each with 3 carbons. The second step is oxidation. So the above chemicals are then involved in a combined oxidative phosphorylation reaction. Hydrogen is removed from triose phosphates, and NAD accepts that hydrogen. The last step is ATP formation. So lastly, the triose biphosphate gives up one of its phosphates to ADP to make ATP and producing pyruvate. Total ATP production for each glucose is 4, but the net is only 2. Here it is in pictorial form. Just like everything else, you need to label a micrograph of a mitochondria. And when you look at this, know that the mitochondria are found in the cytoplasm of all eukaryotic cells in large numbers. It's a large organelle surrounded by an outer membrane with many inner membranes. That inner membrane is folded, and those folds are called cristae. Between the inner membrane and the outer membrane is the matrix. This is a watery, enzyme-filled fluid. It also has some ribosomes and some DNA. The DNA in your mitochondria is maternal, which means we can use it to trace maternal heredity. All right, so the external double membrane of your mitochondria is permeable to pyruvate, so it allows that pyruvate in. It also allows carbon dioxide, oxygen, NAD, and NADH. The matrix is the site of the enzymes for the link reaction in the Krebs cycle. The inner membrane has all of your embedded enzymes for electron transport chain. And your intermembrane space is where the hydrogen atoms accumulate, and then they go through ATP synthase to generate ATP through phosphorylation. Okay, so we have glycolysis has occurred. We've got our pyruvate. Now we're going to do a link reaction. The link reaction links glycolysis to the Krebs cycle. So pyruvate is transferred from the cytoplasm to the mitochondrial matrix, and you see in red the chemical equation for that. The reaction is known as the decarboxylation of pyruvate. Holy cannoli, another big scientific term. Decarboxylation just means you're popping off a carbon dioxide. The aerobic respiration Krebs cycle now is known as the tricarbolic citric acid cycle, or the TCA cycle, or the Krebs cycle. So 3 different names for it. Occurs in the matrix of the mitochondria, and 1 molecule of acetyl-CoA will give us 2 carbon dioxides, waste product, you breathe it out, 3 NADH plus 3H, 1 FADH, and 1 ATP. The NADH and the FADH go on to the electron transport chain, or ETC. So the Krebs cycle, acetyl-CoA combines with a 4-carbon compound that's already in the cycle to create a 6-carbon compound. This compound is then decarboxylated, again, fancy term for popping off a carbon dioxide, and you get a 5-carbon compound. Remember, anytime you pop something off, you also get some energy. The 5-carbon compound is decarboxylated, i.e. popped off another carbon dioxide, and creates a 4-carbon compound that goes back to the start of the cycle, and we go over and over and over. Every time we decarboxylize something, we get some energy. So the main outputs are energy freed from the compounds and carbon dioxide. So note that per molecule that enters, 2 molecules of carbon dioxide are given off, 1 molecule of ATP is formed, 3 molecules of NADH, and 1 molecule of FADH. So here we are. Here's our summary to point with our energy. NADH and FADH are temporary energy storage molecules that are going to transfer energy from Krebs to the electron transport chain. So terminal oxidation and oxidative phosphorylation, fancy terms, but it's called the electron transport chain for short. The removal of the hydrogen from NADH and FADH, those hydrogens are then transferred into the matrix, which then allows us to put it through ATP synthase to make ATP. The end receiver for all of these hydrogens is oxygen. That's why it's an aerobic process. The hydrogen with the oxygen forms water. You pee that out. So for every molecule of NADH that's reduced, 3 ATP are formed, with a general total yield of 38 ATP. Phosphorylation by chemiosmosis. Again, adding phosphates to ADP to make ATP through chemical reactions, chemiosmosis. So the synthesis of ATP is coupled to the electron transport chain via the movement of protons. Electron carrier protons are arranged on the inner folds of the mitochondrial membrane on that cristae. We reduce or oxidize the reduced coenzymes, and the energy from the oxidation is used to pump hydrogen ions from the matrix space between the inner and outer membranes of the mitochondria through ATP synthase. The gradient forms, creating a potential difference. That potential difference is a potential energy storage that we release and generate ATP. Now, glucose is our main energy store, but we can also use fat. So your body can plug other things into the Krebs cycle. It's harder, it's not as efficient, we're not going to get as much ATP. But we can plug in starch, glycerol, fatty acids, and amino acids. Plants can add cellulose to that list, and it's why fat is considered to be an energy source for the body. A couple cheesy songs to get you through. Once you hear them, you'll be singing them the rest of the day. And then, of course, Bozeman, who might explain it clearer than I did. Next up, some baking of bread.